This protocol offers a standardized method for determining immune cell identity and purity and addresses limitation of flow cytometry such as large sample requirement, high user-to-user variability, and subjective data analysis. This assay includes an automated data analysis template which allows for users to assess immune cell identity and purity objectively. This, along with the multiple assay controls, offers a turnkey, easily standardized method for assessing immune cell identity and purity. Demonstrating this technique will be Jerry Guzman, a scientist in our laboratory.
After genomic DNA isolation from the hematopoietic cell sample of interest, use 1 microliter of the genomic DNA to measure the purity of the sample on a spectrophotometer by obtaining the optical density at 260, 280, and 230 nanometers. Next, dilute 400 to 1,200 nanograms of genomic DNA sample to a final volume of 142 microliters with elution buffer from a genomic DNA isolation kit. Add 75 microliters of the calibrator to 67 microliters of elution buffer, and dilute 1,200 nanograms of the reference genomic DNA to a total volume of 142 microliters with elution buffer. Then, incubate the tubes for 5 minutes at 56 degrees Celsius and 900 revolutions per minute in a Thermo mixer. For bisulfite conversion, add 270 microliters of ammonium bisulfite and 90 microliters of THFA to all of the tubes, and vortex the tubes to ensure a thorough mixing. Briefly